Hey guys, welcome back. This is Lei. In the last video, I showed you how to identify viral sequences in regular RNA sequence data. Basically, what we did was to map the reads to multiple viral genomes and count which genome captured the most reads. By doing so, we were able to detect which virus coexists with the host. Once we have this information, we can do some further analysis. In this video, I'm going to show you how to quantify viral gene expression in host tissues. There are two ways to tackle this problem. The first one is the two-step strategy. In this strategy, we map the reads to the genomes twice. In the first step, we map all the reads to the host genome and capture the unmapped reads. In the second step, we map all the unmapped reads to the viral genome and quantify the expression of viral genes. We use this method when the samples have low viral content, or you're concerned about false positives from host viral sequence similarity, or you need high confidence in identifying viral transcripts. The second method is an integrated strategy. We create a combined reference that includes both host and the viral genomes, then map all the reads in a single step. We use this method when you expect substantial viral content, or there's a minimal sequence similarity between host and the virus, or you want a streamlined workflow, and you have very powerful computer. Now, let's start with the first strategy, the two-step strategy. First, we create our viral reference. By doing so, you have to need a powerful Linux system, and we have to activate the conda environment for RNA-seq data analysis we created in the previous tutorial. And we create a directory for the viral reference. The aligner we use here is still star. The virus, example virus here, is the real virus. To create the star index, we need two input files, a genome sequence file in a fast format, and a customized genome annotation file, the GTF file. For the GTF file, here's the table, tab delimit table with 9 columns. You can make this table manually or programmably. So look at the first column is the name of the sequence. CHRUN means chromatin unknown. And second one is species. Third one, just make all exon. Fourth one and fifth one are the coordinates for the viral genes in the genome. You can leave the rest 3 blank. And last one, the ninth one, is the name of the viral gene. So just make sure you have this correct format. Once we have these two input files, we can call star to make the index. So here I use the max RAM. I have a computer with 200 gigabytes RAM. So just adjust it to align with your system. This parameter is important for creating the genome index. For human and mouse, use a large number, basically 14. For smaller virus genome, 6 is enough. And then we specify the input file, the FASTA file for the genome and the GTF file. And here we want to make the index, so specify the output. You don't have to create a host reference, because in my previous videos, you can directly download the pre-built index for star, for human or mouse or for many other model systems. So once we have the genome index, we align our reads to the host genome first. And don't forget to trim the adapters using trim galore. Hopefully you still remember how to do it. And then we set up the parameter for star alignment. Set up the input, the trimmed FASTQ file 1 and 2. Every parameters are same as we showed you before, except this one. We want to capture the unmapped reads and save them to FASTQ files. So in the next step, we align the unmapped reads to the viral genome index we just created. Look at the input files. They are in this format. They are in the files called unmapped.out.mate1 and mate2. And all the other parameters are same as we did before. And next, we use the featureCounts to quantify the viral gene expression. The parameters are same. And -A to specify the genome annotation file, the GTF file, -O to specify the output file name. And the last one is the BAM file you get from the above step. And this is the first strategy. The second one is to create a combined reference and map the reads to this combined reference. Now let's combine the genomes. It's very simple. We use a command called cat. It's short for concatenate. We cat the mouse, because we have a mouse dataset, the mouse genome in a FAST format, and the virus genome in the same format, and output, this description sign is the output sign, to a new combined FAST file called mm10 real virus combine.fa. And then we combine the genome annotation file, the GTF file, the mouse one and the virus one into a new combined one. And then we use the combined genome to create a star index, the same way we did it in the previous step. Once we have this combined genome, we align our trimmed FASTQ files. We already trimmed it in the last step to the combined genome and quantify the viral gene expression using featureCounts in the same way, except we use the new combined genome annotation file. And what's the difference between the two methods? Look at the quantification. They're basically same. So in our case, there's no difference between the two methods. So for the convenient purpose, I recommend using the second one if you expect your virus content is sufficient. And this is all I have for today's tutorial for viral gene expression quantification. And for more information and details, you can go to my website and look at the corresponding tutorial. And subscribe if you like it, and I'll see you in the next video.